Hello, people. Welcome back to my YouTube channel on pharmacognosy-related matters. My name is Judith Constantine from Mohas, Dar es Salaam, Tanzania. I facilitate pharmacognosy-related subjects as well as pharmacognosy-related research. Here we have plant tissue of diagnostic importance. If you have not watched part 1 of plant tissues of diagnostic importance, please watch, because that is the introduction section and it has the issues on the epidermal tissue system, and this is the continuation of what we did last time. So welcome to part 2. Feel free to comment, and please make sure, if you can, please subscribe, share, drop your comments, discussion, and everything that you may raise here. If you remember, this is where we ended last time. We have seen all these epidermal cells with some inclusions and some changes in the stomata, for example, the sunken stomata and the like. And please read through the Trees and Evans book. If you don't have one, make sure you purchase one for you to be conversant with pharmacognostic activities. So we ended up with the epidermis, and we said that the epidermis has trichomes and stomata. And today we are going to start with trichomes in this section. Then we will chip into the stomata in part 3. So trichomes are hair-like structures that reduce water loss and deter pests in plants. Trichomes are highly significant diagnostic tissues in pharmacognosy. They serve as crucial botanical markers for the identification, authentication, and quality control of medicinal plant materials. Their unique morphology, their types, their presence or absence, and distribution patterns are stable characteristics even in fragmented or powdered crude drugs. So their stability makes them good or best or crucial diagnostic tissues in pharmacognosy. So there are two types of trichomes. We have non-granular trichomes, also known as clothing trichomes, and we have granular trichomes. We are going to see these and how they help in diagnosis of medicinal plants. So non-granular trichomes or clothing trichomes, they are primarily serve as physical and physiological protection against herbivores, pathogens, water loss, and ultraviolet radiation. They have varied forms. They can be unicellular, they can be multicellular, they can be branched, they can be unbranched, and they provide distinct microscopic markers. When we say unicellular, these are single elongated cells sometimes, and when we say multicellular, they occur in groups more than one cells. So we are going to see as we move on. Multicellular can be uniseriate, can be multiseriate, can be biseriate. So please go and read through these terminologies. And here we have examples of non-granular trichomes. For example, up here we have unicellular. They have single cells which are elongated. And we have multicellular. You see here we have more than one cells in a single trichome. And another one here, 1, 2, 3, 4. We have many cells in one. And here we have multicellular but branched. For example, here 1, 2, 3, 4. They have branches. We are going to see examples as we go forward. Granular trichomes are characterized by secretory head, often multicellular, and a stalk. They have a head and a stalk, unlike non-granular trichomes, which are just like sticks. The granular trichomes produce and secrete the phytochemicals of medicinal importance. Their morphology and the chemical composition of their exudates are key diagnostic features for many medicinal herbs. For example, the granular trichomes can be found in Artemisia annua and Cannabis sativa, where they produce. In Artemisia we get artemisinin, and in Cannabis sativa we get cannabinoids. So this is how they appear. We have the head which has secretory cells, and in here we secrete phytochemicals, and we have the stalk here. You see the difference between non-granular and granular trichomes? We expect to have the head with secretory cells and a stalk. But here we have non-granular trichomes. They do not have heads, they're just hairs. That's why they are called clothing trichomes. And this glandular can be multicellular. We say that the heading is mostly multicellular, but not always. Yeah. These are examples of trichomes. These are epidermal cells. We said you cannot speak of epidermal cells. Sorry, my battery is low, so connect. Thank you. We cannot speak of trichomes and stomata without speaking of the epidermis. So these trichomes are in epidermal cells. We can see epidermal trichomes here. For example, remember we said something about papillus epidermis in coca. This is an example of a papillus epidermis. But also here we have unicellular hairs. This is a unicellular hair, and this is a unicellular hair, and unicellular hair of them. And we have a cystolith that appears together with a papillus epidermal cell. This is a cystolith in cannabis. It has papillus epidermal cell, but it appears with inclusions. That's why we said we are going to study epidermal cells along with the inclusions. So please visit the book of the 16th edition of Trees and Evans. If you don't have one, make sure you have it so that you will follow along. So that is it. And for example, here we have unicellular hairs from Hamamelis leaf. These are unicellular hairs. And here we have, these are multicellular, 1, 2, 3, we have multiple cells. These are multicellular trichomes that appear together with within the epidermal cells. Also, we have glandular hairs here. We have glandular hairs from this glandular hair from Atropa belladonna, and this one here. We have this from Datura stramonium. We have this from Digitalis purpurea and many other. For example, we have this from Artemisia, and we have these ones from cannabis. So make sure you just study, read along, and you will come together. We have multicellular labiate glandular hair, this one here. So I hope you'll be conversant with these as we move. Now, speaking of the stomata, these are microscopic. So we are done with the trichome part of the epidermis. Now we are going through the stomata. The stomata are microscopic openings formed by guard cells for gas exchange. Remember in part 1 of plant tissues of diagnostic importance, I said something about guard cells. Guard cells are specialized epidermal cells that help opening and closing of the stomata. These are epidermal cells. And I said something about subsidiary cells. These are subsidiary cells. They are also specialized epidermal cells that come along with the guard cells to help in closing and opening of the stomata. And these are typical epidermal cells. These are epidermal cells that have not been specialized to perform any function apart from that of normal epidermal cells. So the shape of stomata, the size, the number, and the index of stomata are crucial diagnostic features for leaf identification. And we are going to see how these appear and what they tell or what they mean when we see them in our samples. So in pharmacognosy, the stomata serve as critical diagnostic microscopic features for identification, for authentication, and quality control of crude drugs derived from leaves and young stems. Be careful with this. I said this earlier, that the epidermis and the stomata are crucial for identification of plants of medicinal plants from leaves and from young stems. The specific characteristics of stomata serve as diagnostic tools that help in establishing pharmacopoeia standards and detecting adulteration. If you don't know about pharmacopoeia, please go and read what a pharmacopoeia means, but it's just a book that has a lot of information regarding certain medicinal plants, and I will share a certain link to a book that will help you have common understanding or widen your understanding on this, on a pharmacopoeia and what exactly it contains in it. So the stomata characteristics which we are going to discuss in this topic are the type of stomata, the stomata index, the stomata number, the stomata distribution, and stomata morphology. Of course, the distribution and morphology are additional. The main issues to discuss here are the type, the index, and the number, but we will do an additional thing about the distribution and the morphology. So we are going to do the types of stomata, of course, in the next part. That is part 2. I mean part 3 of plant tissues of diagnostic importance. So please don't miss this very exciting part about the types of stomata. We are going to see how easily we can identify, or we can identify the types of stomata by just looking at the number of subsidiary cells and the way they are arranged around the stomata. So please don't miss part 3 of this exciting subtopic. Thank you, and see you in part 3 of our topic. Ciao.